I'd like to first thank the committee for inviting me. It's a really big honor to come and talk to you today. And I'm really excited to be the second person to talk to you about bugs today. And that's because insects have really played an important role in the study of microbiomes. Since they're really small and you can catch them and hold a lot of them and you don't have to have approval to do anything to them at all, they've been really, really useful for a lot of microbiome studies done so far that are just sort of now being caught up in mammals. And what we've really seen so far is a lot of associations like Seth talked about before. In aphids, where you have Buchnera that's vertically transmitted, Wigglesworthia, and a lot of Wolbachia. And this is called vertical transmission or maternal transmission. And I think Seth also mentioned this. When you have vertical transmission and mother has a baby, the symbiont or the genotype of the microbial composition is going to be really, really similar to what was in the parents. And it's really tightly linked in terms of coevolutionary terms. In contrast, in horizontal, you can have a symbiont or a microbiome within the parent, but when its offspring are born or hatched, they're usually born or hatched without any bacteria present, or if they are, it's a lot lower than what's in the vertical. And they have to acquire their bacteria or their symbionts from the environment. And the really interesting part of this is they now have a lot of options for what they can acquire. And you might see that they get the same thing that was in their mother or their parents. But they could also pick up an alternative genotype, which may be good or it may be bad. They can also pick up maybe a pathogen, or they're also open to these other non-beneficial bacteria. And this may be bad for the insect host, but it gives us the chance to look at a lot of different things that we can't look at in vertical associations where there's a really tight link between host and symbiont. Here we can separate the host and the symbiont, and we can see the effect of alternative genotypes on the association. So why does this specificity matter? Who cares if you get a slightly different bacterium in your gut? Well, Seth told you why this morning, so I'm glad that I don't have to go through all of that, but there's also a really pretty classical example in plants that have these nitrogen-fixing nodules with nitrogen-fixing bacteria in them, where if you see a plant that has basically its optimal partner, they grow up very well, they're nice and healthy, but if this plant acquires alternative bacteria or mixed infections from the environment, they can have really strong fitness costs, so they can either grow a lot slower, grow not very much at all, or have reduced reproduction. And this is just one example, this paper that was done in acacia trees. So getting the right microbes and knowing what you're supposed to have and what the specificity of your microbes are is really important. So I'm really interested in how is specificity between host and microbe established and determined in these horizontal associations where you have basically all the bacteria in the environment to choose from. And I'm going to talk to you today about some stink bugs, and I'm going to follow this general overview of horizontal transmission. And this is pretty much taken from some models in squid vibrio, where you see out in the environment, the non-host environment, a lot of different microbes. It's basically the soil, which is what these stink bugs live in, is one of the most microbially colonized regions on Earth. So you can see lots and lots of bacteria, and as they become acquired by the stink bug and move through the gut, they become more and more specific. The bacteria are then transmitted back into the environment where they come into contact with all of the other bacteria and microbes in the environment. And I'd also like to point out that this part right here is also really important in determining specificity because there's that benefit. We know that hosts frequently benefit from microbial associations. And there's really a evolutionary selection pressure to maintain that specificity. So it's not just an ecological process where you see a host out in the environment looking for its bacteria, but there's a maybe not co-evolutionary, but a selective pressure to maintain this sort of specificity due to a benefit that might be received by the host or the bacteria. So first I'm going to talk to you a little bit about the environmental process, and then I'll move into the hosts. So in bugs, like I mentioned before, there's a lot of well-known systems that have vertically transmitted symbionts, but in the last 10 years or so, there have been a lot of insects that have been found to have horizontal transmission through a variety of different mechanisms. So I've talked a little bit about environmental determination, basically what's in the environment you live in is what you get, but there's also a lot of specialized transmission mechanisms, such as capsules with symbionts left behind for the eggs, or coprophagy, or other ways of inoculating the site where eggs are laid or hatched. But today I'm going to talk to you about a specific group of bugs called the stink bugs. And you can see here that they have all of these different mechanisms for acquiring bacteria from the environment, but mostly I'm going to talk about ones that have just environmental determination, none of these other more specialized mechanisms. And the bacteria that they pick up as a symbiont is Burkholderia. Burkholderia, as we just talked about a little bit, Rachel told you, is a really interesting organism because it can live all over the place. So I'm going to talk to you today about it in bugs. Rachel just told you it can live in some plants, but it's also one of the major pathogens that's found in cystic fibrosis patients, and that's actually what causes a lot of mortality or morbidity in that disease. It can be found in the soil, in water, and it's also one of the more rare bacteria that can be present in the root nodules in a variety of different plants. So basically this microbe can live anywhere it wants, and we want to know why it associates with insects or what it's doing in there. And so what is it doing in there? This is a picture of a midgut of one stink bug that I've studied. And there are these specialized crypts at the bottom of the midgut that seem to do nothing for digestion or nutrition acquisition. They seem to only be there to hold bacteria. And these are at the very end of the midgut. You can see that this is blue throughout the rest of the midgut. That's not a natural color. We dyed their food blue so we can see where the food is going. And it also makes it easier to do dissections. You can see that food doesn't even get into these crypts. Basically, it all disperses out into the hemosyl before that and is then recollected by the hindgut. So this little area is not really included on the route of food digestion. It's really separated, and it seems to be really specialized for holding bacteria. So the first thing I want to talk to you about is a group of broad-headed bugs in this larger group of stink bugs. And I'm first going to show you some preliminary stuff we did in one species called Elytis. And like I mentioned before, one way that the specificity of host microbe interactions can be selectively maintained is through a benefit. So the first thing we did was test to see, is this a beneficial relationship? As one of the other speakers mentioned, a lot of people assume these are mutualisms just because that word is cool, I guess. But it's really important to see if there's a host benefit and a microbe benefit. Some of the first experiments done with these bugs found that this symbiont is acquired from the soil, so Burkholderia is all over the place. But the soil seems to really be the reservoir for the insect host. That was found by raising bugs in a sterile environment with and without their parents, and in this natural environment with soil or with plants, and it was only under this natural environment with soil that they picked up the microbe. So we did a fitness test where we gave baby bugs, little nymphs, either soil that was just totally normal from their collection habitat, so we're assuming that it had the whole complement of what these bugs are normally exposed to in the environment, including Burkholderia. And in the negative control experiment, we gave them the same soil that had been autoclaved, just to make sure that if there was any nutrients or moisture in the soil, that we weren't depriving them of that. We raised these bugs, which are sterile at this stage, in cages, and then we PCR'd for Burkholderia once the experiment was over, to make sure that this soil treatment was an effective vector of Burkholderia. Before we did that, we measured development, survival, size, and a whole host of other fitness proxies. And what we saw was there was nearly identical development time in both Burkholderia positive and Burkholderia negative insects, but when insects had Burkholderia, they did have improved survival, which can be a big deal if you're a bug that only lives a couple of months and can only reproduce for a few weeks of that. So Burkholderia is definitely beneficial to at least one host that we looked into in this environment. We still don't know much about the symbiont, whether they get a benefit or not, and I'll talk a little bit more about that at the end. So now this was the one this fitness experiment was done in. I'd like to talk about these other 3 bugs that live sympatrically with this insect host and talk more about how their microbiome is determined based on the environment and host factors. So the first thing we did was look at these 4 bugs. Basically, since they live sympatrically, the idea was they're going to be exposed to the same pool of symbionts. So if there's difference in host factors, we'd be able to see a difference in bacterial community in these insects. So what we looked at with Burkholderia is that we found that most of them fall within these 3 clades of insect symbionts. But there are some diverse bacterial strains that we found in these other clades that, unsurprisingly, host soil-dwelling and environmental bacteria, but also have some pathogens in the Burkholderia cepacia complex. After we surveyed just the hosts, we went back and looked for the same bacteria in the environment. So these bugs all live sympatrically, in Georgia at least, and they live on this plant called bush clover, which grows all over the state. The DOT actually uses it to sterilize banks near the highway because it has such a deep root, so it's all over the place. And these bugs live on it and feed on it, and it's, if not their only source of food, it's the most dominant one. So we sampled, in addition to the bugs, some of the soil around basically the bulk soil around the plants. And we also specifically looked at the plants and in the root nodules to see if these bacteria, which sometimes can be associated with legumes, were also in this plant. And we found also a really wide range of bacteria in the Burkholderia associated with the environment. So we found some soil, some plant in this typically insect symbiont clade, but we also found soil all over the genus, which is not very surprising. In the plant nodules, we mostly found rhizobia, which is not very surprising, but this one sequence that we did get from a root nodule that was Burkholderia was nearly identical, at least in 16S, to some of the insect symbionts. So that's really interesting in that we're getting a lot of Burkholderia across the genus, but I'd like to look at this a little bit more quantitatively. So when we looked at all these samples, we saw that the sequences in the Burkholderia insect clade was really the main OTU that was present within these bugs. So it was in all of the hosts across all the sampling we did, suggesting that it is really the most desired or the preferential symbiont. And beyond that, there was a lot of variability. So in each species, we got some different ones scattered throughout the Burkholderia symbiont clades. We also found some of these in a few species, and Cupriavidus, which is a Burkholderia sister genus, was also present throughout there. So unsurprisingly, since they had all this preferential strain, there was no difference between hosts in Burkholderia symbiont community. We did see, though, that collection location had a significant difference between all of the 5 places that we collected them from. And what the pattern really suggested was the environment didn't really affect this main symbiont that was being acquired, but these more rare genotypes were really environmentally dependent. So in some environments, we saw each of these strains in only one environment. So probably that pattern is being driven by the more rare or less prevalent Burkholderia in the environment. When we add the other environmental samples to this, you can see that it looks really different from what we saw in the bugs. So up here is mostly the insect symbionts, and we only found 2 strains, one in the plant and one in the soil, that falls within that group. And most of the strains for both of those environments were down here, unsurprisingly, in the plant and soil groups. So what that's really suggesting is these groups of symbionts that we see in the bugs are not highly prevalent in the soil, and the bug is probably really enriching for them, or at least that they're are quite different from what's in the environment, in the non-host environment. So there's probably not a lot going on in the environment in terms of the dominant symbiont type. Definitely has a role in what the rarer, less prevalent ones are, but we don't really know if that's really quite important in the symbiosis. We did find that there's some Burkholderia in the Lespedeza root nodules, and that's not to say that these bugs acquire the bacteria directly from the roots, but maybe that this plant keeps a reservoir of bacteria that makes them a little bit more abundant or available for the insects to acquire, since they're at such low abundance in the soil. And largely we found that the host determines what symbiont is present in the crypts. So I'd like to move on and talk about those host factors a little bit more and where exactly along the route of acquisition this specificity is being determined. And for that, I'm going to switch to a slightly different bug. This is a squash bug that's also a stink bug that's a relative of all of the broad-headed bugs. As the name suggests, it lives on squash. And it's actually a pretty bad plant pest for crops throughout the US. So just so you know, their Burkholderia falls within the same symbiont clades that I showed previously. All of these circle icons are sequences from insect guts, and that's basically where we see this Burkholderia falling. So this is a really similar insect and bacterial community to what we were seeing before. So I used Illumina MySeq to sample across their whole midgut, which is their whole route of acquisition. Once bacteria are acquired from the environment, they move down the midgut into the crypts. And I used 20 individual bugs, and I sampled the crypts from each of them. So there's 20 crypt samples. And then from each bug, I also took one other section of the midgut. So there's in total 5 different samples of each midgut part: the M1, the M2, the M3, and this little tiny M4B, which is sort of just a tiny little junction between the crypts and the rest of the midgut. What did we see? This data is hot off the machine, so I'm really excited to show it to you. We found that Burkholderia is present in all of the midgut sections. So that's this purplish blue color. This is starting at the top of the midgut, moving down. You can see there's a little bit here, and it sort of increases in prevalence as you move through the midgut, which is really pretty consistent with that model of it comes in, it's not very prevalent, and it can grow as maybe as the gut changes in environmental parameters like pH, or maybe as the host is doing more active selection or screening out of to get this bacteria. Oh, I wanted to point out also, this is absolute abundance. So the only reason this bar is really, really high is because there's more samples for the crypts, but they were mostly Burkholderia. So I wanted to look a little bit more closely at the Burkholderia to see, is this one Burkholderia, or are we seeing a bunch of different strains again? So I clustered all of the sequences just that were identified as Burkholderia and graphed those. This graph is very interesting. That's because more than 99% of all the sequences belong to one Burkholderia OTU or Burkholderia species. And all of these are not zero, they're like 1 to 15 sequences fell within these other OTUs. So that's suggesting, at least in the 16S gene, that these bacteria are all very, very similar, that may be hiding some more important phenotypic or genomic variation, but it seems that these insects are picking up very, very specific strains of bacteria. And I too have some ordinations to show you. This first is colored by bug, so each one of these samples is a different bug that has the crypts plus another midgut section taken from it. And you can see this doesn't really show much of a pattern at all. There's a lot of colors all over the place. But showing you the same ordination plot by midgut region, you can see quite a bit more of a separation where all of the crypt samples are here in blue, and they're pretty separate from the rest of the midgut. This is the whole bacterial community from the mycetes, so that's not too surprising since these mostly had Burkholderia in them. This pattern is probably being driven by Burkholderia presence. But it really suggests that the crypt has something special about it, either an abiotic factors or something the host is doing that really selects a community that's a lot different from the rest of the midgut. So it looks like host screening is really specific. And since we saw the same strain of Burkholderia throughout the whole midgut, it really suggests that this screening from all of the environmental isolates to this very specific strain is happening somewhere before the midgut. We know it's not happening in the environment, so that really suggests something interesting is going on either in the mouth parts or in the foregut before the midgut. And our next step is to kind of take a look at those and see if there's still that very, very strong specificity or if there's more microbes getting into to the host that way. So I'd like to talk a little bit more about another aspect of, another aspect of host screening. The mycetes data was really about acquisition. What's happening when you're first getting your microbes along that whole route of acquisition. But after you acquire your microbes, you also have to keep them happy and keep yourself happy. And there's a level of maintenance that has to happen so that you keep that specificity and that you keep those specific microbes from kind of getting out of control and turning virulent. I would like to talk a little bit more about that now. Unsurprisingly, we hypothesized that the immune system is going to play a role in regulating these populations and making sure that this specific microbe is the only one that remains present in the insect. So we did an experiment where we tested to see whether these Burkholderia were susceptible to host defenses in the hemolymph or the insect blood compared to other strains of non-symbiotic Burkholderia and other environmental microbes. So what I did was first inject LPS, which is a component of the bacterial cell wall, into the insects. And that's just to stimulate an immune response because insects don't constitutively express a lot of their immune components. I let them wait 24 hours in the incubator, and then I collected their hemolymph and put it in plate wells that were embedded with the bacteria, so the different bacteria. If there was an effect of the immune components on the bacteria, you could see a clearance zone, suggesting that it killed the bacteria or kept it from growing. If there was no effect, there was just growth right up to the well with the hemolymph in it. So the first experiment I did was using LPS derived from E. coli and also LPS derived from the symbiotic Burkholderia that was heat killed. And here this graph is showing the radius of an inhibition zone. So these large bars that you see against E. coli suggest that there are immune factors in the insect blood that cleared the E. coli very strongly. So there's something that is killing them or keeping them from growing. However, this symbiotic Burkholderia, which is a strain from the Crypts, there was no effect whatsoever. All of the plates look like this, suggesting that either the Burkholderia can do something to overcome these host defenses, or the hosts are really tolerant towards them. I looked at a few different bacteria that we collected from the environment in the previous, that I showed you previously, to see if this effect was general or if it was just really specific to Burkholderia. And what I saw was really a mix of different things. So again, we saw a really strong response against E. coli. And this Burkholderia in the Crypts was the same from the previous slide. I also included 2 other Burkholderia species. This is the one from the plant root nodule. This is one from the soil. And sort of there's this confusing pattern where there's no effect against the Burkholderia from the soil, but there's a sort of intermediate clearing of the Burkholderia from the plant. So this could help establish specificity, but it's not clear that it's just tolerance towards the symbiotic Burkholderia in the insect. We also looked at one other non-Burkholderia species that we found in the Crypts. This one's a sister genus to Burkholderia, and this one's a totally separate phyla of bacteria. And again, we didn't see a very strong reaction to it. So this may suggest that there's some immune tolerance towards Burkholderia, or these bugs are just crappy at mounting an immune response. So there is tolerance towards Burkholderia. We don't know if it's super specific to that genus or to the symbiotic Burkholderia. So now I'd like to talk to you a little bit more about what happens next after you're a bug, you've acquired these specific microbes, you've nurtured them and fed them, and now you have died. What happens to the bacteria that are in your gut? Do they go back to the environment? And can they be acquired by your next generation of offspring? Transmission screening. For this experiment, we tracked GFP-labeled Burkholderia, which you can see here in the Crypts. It's super heavily colonized. We fed bugs during their acquisition phase a little droplet full of these GFP-labeled bacteria, and then we followed the bacteria throughout the lifespan of the insect. And we looked at any chance that there was for these bacteria to be expelled, so during molts, in poop, and we also looked in the dead carcasses. Once the bugs had died, we left them in the soil to see if there could be transmission back to the soil. Oh yeah, if you don't know, frass is a word for insect poop. My advisor would not let me put the word poop on the slide, but that's what it is. So we sampled some from juveniles, from adults. We swabbed their mouth parts and their feet to see if we could find GFP-labeled bacteria there. This was their molts, and finally their carcasses that we left in soil. And basically what we found was symbionts are not leaving these bugs, which is really quite surprising. It's assumed in the microbiome community a lot that what's in the feces is reflective of what's in the gut. But in the juvenile feces, we didn't find any bacteria getting out at all. We found just 3 out of 73 in adults. And even in the carcasses that were left in soil, we didn't see any bacteria getting out, even though the bacteria was still alive in the carcass. So in some ways, this isn't surprising, because in our initial survey, we found that this strain is not really present in the environment. And these results suggest that that's because hosts aren't really seeding the environment with the strain. They may just kill all of their bacteria once the host dies, or it may take longer for the bacteria to get out than we tested in this carcass experiment, or there may be some other ecological factor that helps them get out, perhaps like predators that break up the carcass, or rain, or different sorts of things that could speed up decomposition of the body. So looking at this whole route of acquisition, we have this model now for these bugs that looks like these Burkholderia aren't really very present in the environment either before acquisition or after they've been acquired by a bug. And there's not a lot of specificity for them in the environment. And where these insects are really getting their specificity is somewhere after the Burkholderia have been acquired. It looks like it's already established in the midgut, so it likely happens somewhere in the mouth parts or in the foregut of the bug. And that's where we're really looking at next to see what's going on in there bacterially. And I'd also like